my name is Zachary. Over the summer, I worked at New York University Medical Center under the lab of Dr. Thomas Wisniewski. The title of my project was Novel Immunotherapy for the Prevention of Amyloid Beta Plaque Formation in Alzheimer's Disease Model Mice. So, Alzheimer's disease is the most common cause of dementia, accounting for about 60 to 8% of all cases. There are three main hallmarks of Alzheimer's disease. The first is the accumulation of amyloid beta plaques in the oligomeric form. What this means is that the amyloid beta protein, which occurs normally in a non-Alzheimer's disease brain, occurs in a large quantity and with toxicity in an Alzheimer's disease brain, as can be seen in the difference between this sketch of a normal neuron and an Alzheimer's disease neuron. As you can see here, the protein has accumulated in a plaque form. The second hallmark of Alzheimer's disease is neuropilin tangles of the tau proteins. In other words, the tau proteins tangle among themselves. This can be seen in the difference between, again, the normal brain and the Alzheimer's disease brain. The third hallmark is cognitive loss, which is commonly characterized as memory loss. The first two hallmarks generally lead to cognitive loss. The mice used for this experiment were Alzheimer's disease transgenic mice. Human Alzheimer's disease genes were inserted into the mouse genome, causing for the overproduction of the amyloid beta protein. These kinds of mice were used because they accurately simulate Alzheimer's disease in humans. Throughout the treatment, behavioral testing was used to determine that the treatment was not affecting the mobility of the mice, which would add a variable into the radial R maze staining. The radial R maze testing. Some of the mice were injected with the treatment monthly for about 9 months. Others were injected with a vehicle solution for the same amount of time. A radial R maze, as mentioned before, was used to test the cognitive function of the mice. The maze was made up of a center platform connected to 8 arms. At the end of each arm, there was a reward for the mouse, which was water in this case. The mice were allowed to stay in the maze until all the rewards had been consumed. Time in the maze was recorded, and the amount of errors were recorded. An error was defined as when the mouse entered an arm that had already consumed the reward. To find the completion of the treatment, the brains of the mice were taken and stained with antibodies for amyloid beta levels. As can be seen here, this is an artist's rendition of the radial R maze. As you can see here, the mouse is heading towards one of the rewards at the end of the arm. The immunotherapy we used was designed to reduce the amount of amyloid beta plaques by attacking a gene that was allowing for the overproduction of the protein to occur. The effectiveness of the treatment was assessed using the radial R maze and brain staining. This graph shows the amount of errors made by each testing group over 13 days of testing. The red line, which can be seen here, represents the treatment group. The blue line, which can be seen right here, represents the vehicle treated group. And the black line, which can be seen right here, represents wild-type mice, or mice that don't have Alzheimer's disease. It is clear that the treated mice made significantly fewer errors than the vehicle-treated mice. This significance is displayed by a p-value of less than 0.001. This image right here is a microscope image of a vehicle-treated mouse stained for amyloid beta proteins. The image to the right of it, right here, is a microscope image of a treated cortex, also stained for amyloid beta proteins. It is clear that the treated cortex has far fewer plaques than the vehicle-treated cortex. This is also represented by the graph to the right and the p-value, which is 0.047. All right. This image right here is a microscope image of a vehicle-treated hippocampus, which is the part of the brain most commonly affected by Alzheimer's disease. And it is, of course, stained for amyloid beta proteins. The image to the right of it, right here, is a microscope image of a treated hippocampus, also stained for amyloid beta proteins. It is clear from comparison and the graph to the right that the treated mice have far fewer plaques than the vehicle-treated. There is a 60% decrease in plaques in the hippocampus and a 64% decrease in the cortex. The p-value of the hippocampus stainings was 0.0013. The treatment clearly demonstrated an ability to lower the amount of amyloid beta plaques in Alzheimer's disease model mice. Due to the success of this experiment, we could hopefully repeat the experiment in the future and eventually find suitable treatment for Alzheimer's disease in humans. I'd just like to thank Dr. Armstrong, Dr. Cox for assisting me with the presentation overall. And I'd also like to thank my mentor, Dr. Thomas Wisniewski, for allowing me to work in his lab over the summer, and Dr. Henrietta Schultzover for allowing me to use the results of the experiment in my presentation. Thank you.